Hi, well, I'm really happy to be here, share with you some of the work we've been doing looking at diversity of cell types in human brain, which is relatively recent work. You heard quite a bit from Vilas, gave a really great introduction to some of the dissection of cell types in rodent, but it's kind of interesting to obviously see what's going on in human. So we know from many decades, over 100 years of work, and probably much before that, that there are a diversity of brains across species. Looking at morphology of individual neurons, but also just the gross morphology of brains. And so it's really an open question now across mammals, how conserved are these cell types, both within a species in different cortical areas, but obviously between species too. And we'd like to profile in human these different cell types, and it would be nice to use sort of precisely the same procedure we did in mouse. It's been very successful, but one of the challenges is that when you are trying to in order to profile individual cells, you need to dissociate them, and it's much easier to dissociate a 2-month-old rodent brain than it is to dissociate a 30-year-old or older adult human brain, where the processes and neuropil is really densely packed, and so you'll see. So I'll talk a little bit about that, and in fact, rather than trying to profile the messenger RNA in the entire cell, we found and others that we're able to get enough information from the nuclei of the cells, and in that way we can profile in a less biased way the different cell types in human brain. In mouse, where we are able to dissociate whole cells and nuclei, that provides a nice platform to be able to really compare the two. Obviously, there are transcripts that they're transcribed in the nucleus, but then are shuttled out into the cytoplasm, into the rest of the cell, and so there will be some differences between the two. So I'll talk a little bit about what those differences are and the expression patterns that are found in sampling the whole cell versus just the nucleus. And given that we're sampling just a subset of those genes in the nucleus, does that change our ability to discriminate different cell types on the basis of their transcriptional profile? And so I'll talk about that in this next section. And then hopefully I'll convince you that there's enough information in the nucleus to actually do this and perhaps get a good enough information to profile in humans. So I'll talk about 2 projects. One was a pilot study to look at diversity in 2 different cortical layers that have a diverse number of cell types as described by their morphology, layers 1 and 5, and then sort of the first steps towards building a real reference atlas that will describe cell types in all different layers in a particular cortical region that will then be publicly available for everyone to use. So first off, I just want to really drive home that brains vary greatly in their size and shape across species. I think we're all kind of aware of this. Obviously, a mouse is much smaller than a human, so it's going to have a smaller brain, but the shapes of these brains also differ. And one other thing to note is that very large brains have evolved independently on diverse branches of this evolutionary tree. So you can see in the top left the sort of elephant brain, we're sort of in the middle there, a slightly smaller brain, and dolphin in the lower right. And so there's been really this real diversification of different brain shapes and sizes. And so what might be driving that? Part of that is the adaptation of species to their ecological niches. So for example, a tree squirrel is jumping from limb to limb of a tree and really needs to have high visual acuity, needs to be able to process visual information very quickly or it's going to fall on the ground. So as many of you I'm sure are aware, the cortical sheet is not a completely uniform structure. There are inputs that are coming into different areas from different sensory organs, for example. And so the primary visual cortex, which Vilas already introduced in the rodent, is expanded in the squirrel, but in a species that relies much more on sensation, for example, the platypus, where it's in very murky water and visual cues are not quite as useful, has an expanded somatosensory cortex. And likewise in bats, they obviously using echolocation in the dark, and so they have this expanded auditory cortex. And so, over evolutionary time, these different functional areas have enlarged or contracted. There also have been larger brains tend to have more of these cortical areas. There can be specializations within areas. And so really it's farthest thing from just being a sort of homogeneous structure. Then if you zoom in at a more micro scale, looking at the histology of different sections of brain, based, now 100 years ago, von Economo and others, and Ramon y Cajal have described a broad diversity of different neuronal morphologies. And at the very least, we know that the relative proportions of these different neuron types, based on their morphology, vary across the cortical sheet. And so this is just a little schematic in the lower left showing by color, different portions of cortex that seem to have different cytoarchitecture, different sort of mixture of these different cell morphologies. And so we know at least that there are different proportions. We also know of more recent work looking at the expression of single genes, showing on sort of the middle of the screen is the in situ hybridization data from the mouse, looking at a sagittal view of the brain, the whole brain. And so this gene post-N is enriched more in frontal areas of the cortex, whereas BMP5 is more enriched in the back of the brain. And so we already know that individual genes show differences across the cortex. Profiling with microarray data, bulk dissections of the monkey brain, we also know that this is true for across many genes, not just a few select genes. But the question really remains, are these really the same cell types, but markers of those types are sort of shifting over the cortical sheet, or are there really unique types that are distinct to these different areas? And moreover, how might these types be shifting across the sheet, the cortex? So on the one hand, work looking at just neuronal counts across the cortex has shown sort of a gradual increase from frontal areas to more rostral areas. So primary visual cortex has a very large number of neurons that are under a square millimeter of cortex compared to frontal. And profiling in human brain, this was work published by the Allen Institute, looking at microarray, these were dissections from human brain from many different cortical regions. And one result from that was showing that two cortical areas that are very close to each other on the cortical sheet also have more similar expression profiles than two areas that are very distant. And that this is a fairly smooth fall off with distance between different cortical areas. On the other hand, other work has suggested more of discrete changes by functional region. And so this other study using a different method of cell counts on the right showed that there really were the highest density of neurons in primary visual cortex and somatosensory cortex, but it wasn't quite in this perfectly graded way. And likewise, even though that there's this sort of smooth change in expression patterns across cortex, the most distinct areas in human brain are also these two primary sensory areas. And so the story is probably somewhere in between these two, but it would be interesting to know, how does this expression, how are these expression changes reflected in changes in cell types? So one idea to keep in mind. So as Vilas nicely illustrated that in his paper they worked on with Basilka and others at the Institute, they identified 49 of these cell types in mouse cortex. But now I'll tell you a little bit about some of the initial work where we tried to use whole cells to perform this study in the same way. And really few studies have been able to demonstrate recovery of these whole cells from the adult human brain. And really this is, when you digest, you have to separate out these neurons some way. So you use an enzymatic dissociation method. And that ends up giving you a lot of cellular debris. And unfortunately, there aren't any very specific surface markers for the cells of interest. And so you're able to sort out some of the debris, but you can't really enrich as much as you might want to for the neurons themselves. And so this was an experiment that Rebecca Hodge and our group did, where we were able to recover some cells from dissociated cortex, but one would expect roughly half of those cells to be glial and half to be neurons, based on known proportions in the cortex. And really what we found was, even with a variety of different dissociation methods, that most of these cells were either low quality or glial. So we got very low recovery of neurons. And that might be good enough, except we might think that we're not really capturing all the neurons. So it was a bit worrisome to go this route. And so in contrast, if we try to sort individual nuclei, so here I'm showing, these are gyri from a postmortem adult human brain, and these are cut into 0.5 millimeter sections, and then dissected, individual layers are dissected out. And then on the basis of nuclear stain, the nu-N, which should stain only neuronal nuclei, we're able to really separate out the nuclei from all the debris, and also the nu-N positive nuclei from the nu-N negative. So shown in the middle bottom here. So these are the nu-N positive and then the nu-N negative. And when we profile these nuclei based on the RNA-seq sequencing, and then look at their marker genes, we find that this study was designed to collect about 10% nu-N negative nuclei, and that's about what we see based on their gene expression profile. And the nuclei isolated from layer 1, we would expect to be more inhibitory. There's enrichment of inhibitory neurons there, and indeed we see mostly inhibitory, majority of inhibitory neurons. And from layer 5, we see sort of this mix, expected ratio of excitatory and inhibitory types. So this was promising that we were at least getting on a broad level the nuclei, the cell types that we would expect. So now I want to switch from human back to mouse, where we are able to compare what we're getting from nuclei to the whole cells. So Vilas already showed you this graphic of the mouse, schematic of the mouse brain. So this now is a dissection just of layer 5 of primary visual cortex. And we're able to isolate about 470 nu-N positive nuclei. And about 85% of those were excitatory and 15% were inhibitory. And the strategy was to find a matching set of whole cells that we could then compare those nuclei to. And so we have sort of the luxury of this really large whole cell data set now. It's expanded greatly from the initial study that was published with 1,600 whole cells. And so we had about 2,500 whole cells dissected from layer 5 that we could then match to these nuclei. And just comparing expression of all genes between individual whole cells and individual nuclei, you can see that for some pairs, there was a fairly high correlation between the two. And so for each of those 470 nuclei, we could pick the best matching whole cell. And so this is the distribution of those correlation values. Most were above 0.6, which is reasonably good. And these whole cells were represented by a variety of different Curie lines from different transgenic mice. So I need to just give you a little bit of a small detail on how we measure, profile the expression levels of these genes. You end up using this procedure with a whole bunch of relatively short fragments of nucleotides that then are mapped to the genome. So this is mapping these reads to the mouse genome. And then from here, there's a whole set of known transcripts where you can imagine two reads might be mapped to distant locations on the genome, but if the genetic sequence in between is intronic sequence, then that's spliced out in the nucleus and when you're making the messenger RNA. And so you end up you wouldn't necessarily think this was the same gene, but since we have these nice models of how these different exons fit together, we can map then from this set of mappings to a transcriptome. But some of these mapped reads, if we don't have information about possible transcripts they might map to, are left sort of just tagged to the genome. And so the reason why I'm going through this is because we see a difference in the whole cell data and the nuclei in the percentage of the reads that are mapping to this transcriptome step and this genomic step. And specifically, many more of the, a higher percentage of reads in the nuclei are mapping only to this step and are not mapping to known transcripts. And so the question is, are those reads sort of junk? Is it that we have so little information in the nucleus that perhaps they're just randomly mapping to across the genome, or are they mapping within the known gene bounds of those genes? And remember, genes only take up about 1% of the genome, and so by chance you wouldn't expect them to sort of land right in the gene. So what we can do is for this percentage mapping to genomic and messenger RNA, we can break this genomic DNA into intronic reads, which again are in between the gene bounds, and reads that are mapping just randomly in the genome. And you can see in the nuclei, many of these reads are mapping to introns. And so there seems to be real information content there. And we're still investigating what these reads might represent. They could represent unspliced transcripts that aren't quite ready to be shuttled out into the rest of into the cytoplasm. They could be partially degraded transcripts. They could be novel transcripts that haven't been described before. In some ways, it doesn't matter for the purposes of defining cell types if it's providing useful information, although we are very interested to explore that more. But I'll just say as a reminder, here's what we're comparing. So when you dissociate the whole cell, you're getting the cell body, the nucleus, and maybe the very first parts of the processes of that cell. But of course, you're losing transcripts that have been shuttled off into the ends of the processes. Whereas the nucleus, you just get the nucleus and maybe a few transcripts that have stuck to the outside. And so you'd expect that you're probably going to detect more genes when you get more of the cellular content than with here than here. And indeed, that's what you see. So in red on the box plots on the right are the number of genes detected for whole cells, and for nuclei are in blue. And using information just from the introns, exons, or using information from both, we see more genes detected in the whole cells. But including these intronic counts really boosts the number of genes detected in the nuclei. So this was an important piece of information that we really need to include, not throw out all the intronic reads. And one of the differences with sort of the analysis of the whole cell data. So as I mentioned, the transcription of RNA happens inside the nucleus. And this happens sort of in a bursty way. So genes are sort of transcribed when a signal comes that gene is needed, that the protein that's created from that gene is needed. But it doesn't happen in a completely smooth way. There's this sort of probabilistic transcription. And then these transcripts sit in the nucleus for anywhere between 5 minutes, half an hour, and then are shuttled out into the cytoplasm where they either are degraded or are used to create different proteins. And the effect of this is that you get, showing in the plot sort of schematic in the bottom, is if you count the number of transcripts for a particular messenger RNA within the nucleus, in blue it's very spiky, but because of this buffering and retention time, you get a sort of smoother distribution over time in the cytoplasm. And so you might think, so sort of the effect of this is that you are basically you expect to see gene dropouts. So genes that maybe in a cell type like a parvalbumin cell type is one example, where really in the whole cell data you see parvalbumin turned on in all of these cells, but because of this sort of bursty transcription, you might expect it would just drop out in some nuclei. You wouldn't see any expression level of it. And in fact, this is what you see for a minority of the genes. So on this plot, I'm showing So this is sort of a density-based plot, so the color indicates how many I'm plotting genes here. So on the x-axis is the proportion of the nuclei where that gene is detected, and on the y-axis is the proportion of whole cells where it's detected. And so you can see most of the genes are down here with very low expression in both nuclei and whole cells, or very high in both. But in general, it's a little hard to see here, but if you add up everything in this sort of central line, more than half of the genes are sort of equivalently detected in whole cells and nuclei. But because of gene dropouts, genes that are up here are detected more reliably in whole cells. And so this is just something to keep in mind as we do the clustering. Just as sort of a sanity check, what are these genes that are more reliably detected in whole cells? So again, these are violin plots. Vilas already introduced these, but just to say, nuclei now are in red and whole cells in blue. And this is the distribution of expression values for each of these genes. And here I'm plotting expression for genes involved in translating RNA, messenger RNA, into proteins, and also mitochondrial genes, which you wouldn't expect to be present in the nucleus at all. So, and indeed, some of these really show no expression in the nuclear data and just in the whole cell data, leading us to believe, this is as expected. They're in the cytoplasm. Interestingly, there are some genes that seem to be enriched in the nucleus and not in the whole cells. So genes involved in splicing the RNA, and so they may be sort of sequestered in the nucleus and then shuttled out and a protein made and then brought back in. So, and then somewhat intriguingly, these calcium channel subunits, which are obviously important for a lot of cell functions, but we're not still investigating why. It's interesting just to note that there are some spatial localization to these transcripts within the cell. So really, at the end of the day, we want to know, can we discriminate different cell types using information from whole cells and nuclei? So again, I'll go through this a little quickly because it's already been introduced, but the general strategy for clustering is selecting variable genes, reducing the dimensionality, and then clustering that data, and then doing this iteratively, where you take subsets of those cells and find new sets of variable genes and cluster those in turn. We can do this with different subsets of the data to really find out the robustness of these clusters. And so here I'm plotting for each cell, for each nucleus, nuclei are along the rows and along the columns, and the color indicates how often that pair of nuclei were put in the same cluster across many different runs. And so you can see there are these kind of tight clusters along the diagonal, and the nuclei are not often shared between clusters. And so from that, you can sort of define your set of clusters. That was an example from a larger data set, but here I'm showing the co-clustering from just this sample of 470 nuclei. So for these, I found 11 non-outlier clusters. 2 of these were inhibitory, 9 were excitatory. And here I'm plotting different marker genes for some of these clusters. So many of these marker genes were known because this is from primary visual cortex. We knew these from the nice study that's already been published. And so this is showing it for nuclei, and then the same exact process repeated for the whole cell data. And in general, you can see the sort of pattern of markers is quite similar between the two. So we can do this a little bit more concretely here, or more rigorously, in comparing how similar are the different clusters to each other based on variable genes across them. And so I built a tree here where you can see that I've now included all the nuclei clusters and all of the whole cell clusters. And for many of these, there are distinct pairs. And so just as an example, this, again, I think Vilas had mentioned this, but this ChRNA6 cluster that's it's a very distinct cluster, and based on the expression of this one gene has very sparse expression in the in situ hybridization data. And we can detect it with both the whole cell and the nuclei data. So this was encouraging. And now, so now I'll tell you about how we applied this to human. So the strategy was to go after these layers where we know there's neuronal diversity based on many years of morphology work and other work. In particular, we wanted to see if the hypothesis was that this very distinct type, this spindle neuron or von Economo neuron, that is very large neuron that sits in layer 5 in very specific regions of human cortex in the frontal cortex, and is really only been seen in large brain social mammals. So this was a particular interest, and perhaps a cell with such a distinct morphology would also have a distinct transcriptomic signature. That was the hypothesis. And then we included sort of this layer 1, where there's a diversity of inhibitory cell types. So this data set included about 1,000 nuclei from layer 5 and 1,000 from layer 1. And again, showing a similar co-clustering plot. You get a lot of clusters. Some of these, there's always sort of always read the methods carefully in these papers, because there's always a step of sort of manual curation, making sure these are real, and you haven't oversplit things. If you don't see very distinct markers between them, you probably want to merge them. But once you've sort of done this curation process, we ended up with these sets of inhibitory, excitatory neurons and a few glia. So again, as I had mentioned, that we expected that layer 1 is really all inhibitory neurons, but we've probably clipped a little bit of layer 2. And so I'm showing sort of the proportion for each of these clusters of nuclei were derived from layer 1 and layer 5. So this one cluster really seems to be a fairly homogeneous set of perhaps upper layer 2 excitatory neurons. And then there's a lot of inhibitory diversity in layer 1, but also some inhibitory types that are found in both layer 1 and layer 5. So there actually is a study now published just this year from Kun Zhang's lab at UCSD on taking kind of a first pass at looking at diversity in human cortex. They used a different platform to measure these nuclei, and they were fairly conservative in how many clusters they reported. But it would still be interesting to see, where do our clusters map on to these published clusters. And so they described these 8 excitatory neuronal types and 8 inhibitory types. And what we could effectively do is map each of the nuclei from our human data set onto this, pin those onto this tree based on the expression of different marker genes. And so I won't go into detail about how we did that, but just to say you can sort of with some reliability pin a nucleus onto this tree. And this is showing how confident we were in that mapping. A majority of them mapped quite well to these different types. And if we look just at those nuclei that seem to map well, we can then compare. We can say, for the clusters we found in our study, which clusters do they map onto in this Lake et al. Study? And so, as I already mentioned, this very large, kind of homogeneous excitatory cluster, mostly from layer 1, mapped onto their sort of layer 2, 3 cluster. There we found a number of different layer 5 subtypes. This makes sense because we really sampled layer 5 more deeply than they did. And then there were a few clusters that didn't map particularly well to any of their clusters, which I'll get to in the next slide. Likewise, for inhibitory types, again showing our clusters on the left and theirs on the right, a few of these mapped fairly closely one-to-one, so we seem to be profiling really that same type, but others we found perhaps additional diversity. So these 2 clusters mapped to this other NDNF positive cluster, and likewise we found a lot more diversity in this type, which again is because we've oversampled layer 1. So we've really captured perhaps more of the diversity in human cortical layer 1. So I told you that these 4 clusters were not mapped. So here are one reason for that is probably because I've already showed you that marker genes vary across the cortical sheet, and And so we profiled frontoinsular cortex, which was not profiled in that study, and so you might expect there might be different cell types in that region. This is showing the marker genes for these different excitatory types, and there were these 4 types that expressed FESF2 and BCL11B, which another name for that is CTIP2. And so this study, this fairly recent study from Bill Seeley's lab, looked at von Economo neurons, the neuron we were really targeting, and showed that they co-express both this FESF2 and CTIP2. So we could narrow it down to these 4 clusters. In particular, this very distinct cluster also express GABARQ and ADRE1A. And by total happenstance, this, not complete happenstance, but this is one subunit of the GABA receptor. And so this we actually had in situ hybridization data from in our online atlas. And this is sparsely expressed in layer 5 in very specific regions of cortex where these von Economo neurons have been described. And so now, one caveat to this is that these don't look particularly spindle-shaped, and so we really need to go in and validate these, looking at carefully at the neuronal morphology. But it's at least promising candidate for a transcriptomic type that really lines up with the, or at least that's the hypothesis, with this morphological type. Also kind of consistent with this, there have been these von Economo neurons described in monkey and in restricted cortical areas, and also GABARQ is expressed also in a very restricted part of cortex. And then there's really no GABARQ expression in the mouse cortex at all. So it would be interesting to dive more into perhaps some of these species differences. The other thing is these neurons, as I mentioned already, are very large. They have 3 to 4 times the volume of surrounding pyramidal cells. And intriguingly, they also have sort of the highest gene detection of all the different clusters. So this could be just, we have higher signal because we get more content out of the nuclei. And in fact, we can go back and look and see, how big are these actual nuclei, and that's something we're planning to do. So finally, I'll just show very briefly how we're now extending this method to other cortical layers. So the strategy was to capture cells of a certain rarity. So you can expect if you had completely unbiased sampling, which probably is not true, but that's the best we can do to assume that, that with these given samplings, given the relative numbers of neurons based on cell counting that are in the different layers, we can expect to capture about cells, at least 16 nuclei, with a 0.2% rarity of all neurons in cortex. And the reason why we targeted that number is because, again, this ChRNA6 type is really quite rare. It represents in mouse cortex about 0.6% of cortical neurons. And so that's really the threshold we were going after. And the eventual data set will include 15,000 nuclei from human cortex. Currently, we have about 3,500 nuclei. This is showing the distribution of their sampling across the different layers. So again, we're capturing about 10% new and negative to profile some glial diversity. And then this is sort of still heavily biased towards layer 5, but we're starting to fill out some of these other layers. We see very distinct cell types coming out of this, which is promising. And then again, as sort of seen in other figures, but if you can relate these different clusters based on their expression profiles, they seem to make some sense. So the sort of inhibitory types cluster together, the excitatory types, and then the different cells that were dissected from specific layers also seem to cluster together. So this is, again, just a heat map view of what you saw with the violin plots showing the expression of different key marker genes. So I've ordered them not by their marker gene expression, which is why this is a bit jumbled. I've ordered them based on their laminar enrichment. But even looking here, you can see somatostatin, a large class of inhibitory neurons, is expressed in a number of these different clusters, and these clusters look quite distinct. In general, they're for GABAergic, inhibitory cell types. There are some that seem to be very layer 1 specific, and then there's a divide between sort of more superficial layers and deep layers. Whereas, as has been described many times and based just on in situ hybridization data from single genes, these excitatory types are very restricted to individual layers. Yeah. So finally, again, as Vilas already mentioned, but we really want to understand these transcriptomic types are sort of putative, I would call them putative cell types. We really need to define a cell type in the context of the circuit. One step moving in that direction is to collect different data modalities from the same exact cell. And so then you can maybe really tie the gene expression profile to the shape of the cell and its firing pattern. And so this is a technique that's been recently optimized, this paper from the Talias lab, where one can patch onto a cell with a glass pipette, measure the firing pattern of that cell, and then extract the contents, hopefully also including the nucleus, for profiling by RNA sequencing. And then one can take, this data tends to be a little bit noisier than data that's from these dissociated nuclei, but there's still enough information to then pin that particular cell onto this tree that we've defined based on this reference panel of types. And so this is the general strategy where you have a tree where you have the different excitatory and inhibitory types, and based on these marker gene expression, we can, for example, pin this cell to a more superficial layer excitatory type and a firing pattern that fits that, and then a PV type fast spiking type where there's, in this initial pass is just really a sanity check that these firing patterns fit what the gene expression would lead you to believe. But I think what will be really interesting is to see overall, as we build out the tree, as we collect more of this data, we can see how well does every transcriptomic type have a distinct firing pattern, for example. So just as just to remind people that we're profiling one or two areas in a very diverse set of functional areas across the human cortex. This nice paper came out just this year looking at the parcellation of the cortex based on structural MRI data, functional MRI data, and other modalities, and defined well over 100 areas in human cortex. And so this is really just the beginning of an understanding of cellular diversity in the human cortex. But I think it'll be interesting to compare these different taxonomies based on gene expression, based on physiology, morphology, connectivity. And again, as I mentioned, trying to understand are types changing in more gradient fashion? Are they changing in discrete ways across functional areas? I suspect it's likely a mixture of both, but we'll see. How do these cell types vary across species as we include more species in these comparisons? And then, ultimately, how can we, once we've understood or at least kind of cataloged this set of cell types and then identified markers that allow us to genetically target those types, then we can really start to ask questions about the function of those types in circuits and how they may be dysregulated in different neurological and neuropsychiatric diseases. So that's really what I wanted to share. There's a huge team of people. Terry's shown this slide, but involved in this. I'm in the human cell types team, so there are a lot of people in that team and the mouse cell types team, and people generating a lot of the RNA-seq profiling from these nuclei, and then the whole information technology infrastructure to sort of track all this data and is really critical. So thanks.